We're going to start the next talk. And our speaker is Professor Kam Leong. And currently, Kam Leong is a Sheng Professor, Department of Biomedical Engineering, Department of System Biology in Columbia University. And Dr. Leong received his PhD in chemical engineering from University of Pennsylvania. And he has been on the faculty of Johns Hopkins University, Department of Biomedical Engineering for over 20 years, almost 20 years. And then he moved to Duke in 2006. And over there, he pioneered the work to study the interaction of cells with nanostructures for therapeutic applications. And in 2014, Kam moved to Columbia University to take his current appointment. And over there, he continues to work on the nanoparticle-mediated non-viral gene delivery and immunotherapy. So in Professor Leong's lab, his main research is on application of nanostructured biomaterials for regenerative medicine. So this is a very timely topic for today's symposium. And specifically, he made a lot of contributions on understanding of cell topology, topography, interactions and applications of non-viral vectors for direct cellular reprogramming and genome editing. And Professor Leong published many papers, many peer-reviewed publications, 320 peer-reviewed research manuscript with very high citation. And on average, every paper is about 100 citations. Total is 33,000 citations, and with more than 50 patents. And Kam also received many, many awards and recognitions, and among them, he is a member of the National Academy of Inventors and a member of the National Academy of Engineering, and currently he's the editor-in-chief for a major journal in biomedical engineering, the Biomaterials. So let's welcome Professor Kam Leong.
Thank you, John. This is truly a pleasure and an honor to be here to share with you what we are doing on how to convert adult cells from one lineage to another, if someone can maybe deem the light so, but that is not crucial if it cannot be done. So the field of regenerative medicine got this excitement in 1993 when three labs in the world simultaneously reported the possibility to get human embryonic stem cells. So these are totipotent cells that can form any tissues in the body. But the requirement of embryo has hindered the progress of that field. And also because these are totipotent cells, they would form any tissues, they would also form teratoma. And so that's another issue that has to be dealt with. In 2006, Yamanaka in Japan showed that it's possible to reprogram an adult cell back to the pluripotent state. So these induced pluripotent stem cells are also totipotent. They can also form any kind of tissues in the body. Now you do not need an embryo. So that has generated tremendous excitement in the field, and for that invention, he received the Nobel Prize in 2012. However, these are still cells that may form teratoma in the body. In 2010, Maris Wernick at Stanford showed that it's possible to directly convert adult cells from one lineage to another lineage without going through the intermediate embryonic state. So now then it's another way of getting the cell source, maybe without the danger of forming teratoma. And there are other features that might make direct reprogramming attractive. And so that is going to be the topic of my talk. So another way of looking at it, the iPSC route of getting the target cell has the advantage of getting expandable precursors, and that in general is more easy to direct this embryonic stem cell into the target cells. In general, cells going through this route would be more functional and so on. What I would like to talk about is this route to go from adult lineage to another lineage without going through this stage. So the characteristics that oftentimes is more difficult to reprogramming cells this way, and the target cells may be less mature compared to this route. However, it does have the significant advantage that one could bypass this step. And also, if one thinks about using this kind of cells in vivo, one may not need to purify the cells because there is much less of a danger of forming teratoma in vivo. So there is significant advantage with this direct conversion route or the transdifferentiation route. So with that, let me show you 3 examples in this talk. The first talk is how to convert adult cells into the smooth muscle cells. The rationale is that SMC play major role in the vascular systems. However, it's not easy to get these cells because they have limited proliferative capacity. So it's difficult to get these cells in large number for applications. It has been known in the field that that myocardium, that this transcriptional activator, that this gene would upregulate the SMC phenotypes of the cells. So we hypothesize that if we overexpress this gene, we can convert cells into the SMC lineage. And in this case, we pick human endothelial progenitor cell as the starting cell source because it's easy to get these cells from blood, and these are also highly proliferative cells, so we can get a large cell number. So very quickly, just to give you an idea of this process, if we overexpress the myocardium gene in endothelial progenitor cells, within a few days we see a significant change in the morphology of the cells. In 1 week, we can see the upregulation of the biomarkers for the SMC lineage and downregulation of the endothelial markers. And we also show that the cells that have been reprogrammed, they are robust. They show the SMC phenotypes and not the endothelial lineage phenotypes. At the molecular level, we show that there is upregulation of the smooth muscle contraction pathway and downregulation of the pathway related to the cell cycle, which is consistent with what we see that these reprogrammed cells do not proliferate as well. And principal component analysis show that the cells are quite different from the starting cell source. And when we go from 2 to 4 weeks, that the cell would get closer to the native smooth muscle cells. At the cellular level, we can see that these reprogrammed cells do show calcium transient, and in using traction force microscopy, we can see that the contractility of these reprogrammed cells would be at least an order of magnitude higher than the starting cell source and could begin to match the contractility of the native umbilical cord SMC. At the tissue level, we can hit the cell, the human endothelial progenitor cells with the myocardium gene, embed them into dense collagen gel, and then coats the lumen of this tubular structure with the same starting endothelial progenitor cells, connect them to a bioreactor for perfusion. In 1 to 2 weeks, we can see the SMC markers in the wall, and we can show the vasoactivity of such a tubular structure that could function like an endothelialized tissue engineered blood vessel. They respond to drug to show vasoconstrictive response to phenylephrine, to acetylcholine. They show vasodilation to flow, show also vasodilation as well as increase in nitric oxide. So this kind of TEBV, we hypothesize, may be biomimetic because of the presence of the SMC. And the interest of this work is on tissue on a chip or organ on a chip. So let me very quickly talk about this concept. There's a lot of interest in the past few years to develop organ on a chip, tissue on a chip for drug development. The cost of drug development, probably many of you know, has gone up exponentially with time. There's actually kind of a crisis right now because of the cost. For any new drug development would be over $2 billion, and the average time is 12 to 15 years. For example, in the past decade, from 2006 to 2016, the LOA of any drug candidate going into phase 1 to the time that it could be commercialized, for oncologic drugs, the success rate is only 5% or so. And for drugs for treating mental health and so on is about 6%. So the failure rate is very high. And many companies are actually cutting down on the investment for the development of these drugs. So there's a big need to come up with a more predictive model. Now, there are many factors for such a high rate of failure. One factor could be just one factor, is that all the preclinical studies done in rodent models, even non-primate models, just cannot recapitulate the response in human. So what is the next best thing that you can do? You cannot test it in human. So the idea is to use human cells to create 3D tissue coupled with physiologically relevant flow for that kind of testing. Now, this kind of model, we are not saying that could totally replace the animal model. I think that will never happen. But with such kind of a model, then will give pharmaceutical companies or even academic researchers a greater confidence of taking any candidate into the clinical trial. And so NIH in the past 3-4 years, maybe 5 years, has this microphysiological system initiative and has funded 18 labs in the country. Now it's 12 labs, and now is in the second phase right now, funding different groups to develop different organs on a chip. The vision is that in the end, that this will all be integrated in such a way that would be useful for drug development. So calling it tissue on a chip, and someone even calling it human on a chip. Now, in this kind of scheme, that the drug would flow from one chamber to another chamber through plastic tubing. But in vivo, drug is exposed to a functional endothelium, which is not incorporated in such a system, we would like to advocate that it would be good to incorporate such a tissue engineered blood vessel into the system so we can more accurately evaluate the toxicity of a drug. So what I've shown you in the tissue engineered blood vessel, the advantage is that it may be more biomimetic. It's known that endothelial cell SMC interaction is important for a functional endothelium. And because this can be made with a single cell source, allowing one to screen against genetic background, age, gender, and so on. So let me just show you one pilot study. In one particular case, we would like to use this to model the metastasis process. So in a tumor bed that because of the cell death, chemotherapy, and so on, would kill a lot of cells and would generate the damage-associated molecular patterns. Many enzymes would go up, would break down the extracellular matrix of the blood vessel, and that would lead to intravasation or extravasation of cells. And so we would like to use this tissue engineered blood vessel to model that process. And right now, the whole field so far has been using this kind of 2D model to look at the interaction of endothelium with immune cells. It's just a transwell model. You grow a confluent endothelial cell layer. You put the immune cells at top. So in time, you would see the cells going through the layer. But this is a static system without flow and so on. Now we can use this tissue engineered blood vessel to model that. This would be the lumen, and this would be the wall of the blood vessel. We label the cells with different color, different label, and now we can model the system. So the immune cells will be flowing through this way. You see here, if you focus on this point, you can see the cell adhering and then disappearing with time. And if you wait a little bit longer, you see another cells adhering to the blood, to the endothelium. And with time, this cell will also disappear because it has gone inside to the wall. So for the first time, we can image this process, how immune cells interact with the endothelium. And so we can quantitatively measure the number of cells adhering to the endothelium and also the number of cells undergoing the transendothelial migration on an activated endothelium. And this may be a possible platform now for drug screening. Here we show that if we inhibit the activation of the endothelium by a TNF-alpha neutralizing antibody or other low molecular weight compounds that block the NF-kappa B pathway, we can inhibit the endothelium activation and can reduce the number of immune cell adhesion and transendothelial migration back to almost the normal level. So that is where we are now. And so just kind of giving you the concept of what this kind of microphysiological system may be used for drug screening. So for the first part of this talk, the summary of that, for this pathway, the direct reprogramming of human endothelial progenitor cell to smooth muscle cell is a fast and robust process with a single transcription factor. It shows good functionality at the cellular and the tissue level. It does have the drawback that this induced SMC adopts the contractile phenotype, but it's not proliferative. And so that's also the reason why we pick the endothelial progenitor cell as the starting cell source, because then we can work with a large cell number. So let me go to the second example that I would like to share with you is to get cardiomyocytes from this direct reprogramming route. Of course, a lot of interest getting these cell types is for therapeutic purpose of myocardial infarction. And so this may be for therapeutic function, also very useful for disease modeling purpose, and also very useful for drug development process. Cardiac toxicity and hepatic toxicity would account for more than 50% of the failure rate of any drug development. So a lot of interest to integrate this kind of organ on a chip, liver on a chip, and cardiac tissue on a chip for drug testing. Now, it's much more difficult to generate cardiomyocytes now. And I would like to preface that other groups have been able to do this, and we have not been able to do as well. But let me show you our effort. It takes more than one transcriptional factor. In fact, a number of genes are required to reprogram the human fibroblasts into cardiomyocytes. There are 7 genes in the literature that have been shown that would be needed, and we group them into different groups and then do a systematic study to see what is the best combination to get this cardiomyocytes. We concluded that transcription factor alone is inefficient and may not be enough, even with the best combination that we can do. Then we borrow from the literature where people do this kind of reprogramming from iPSC, showing that microRNA can also help the reprogramming process. So we also test the combination of transcription factor with microRNA with MIR1 and MIR133A. Indeed, just giving you a flavor, we again do a systematic study on grouping different combinations of them. Now we can begin to see the cells that we would like to see, but it is still a relatively inefficient process. With the 5 different genes plus MIR1 and MIR133A, we can get about 4% of those cells that we need. These cells do show the characteristic of a cardiomyocyte, showing that there is rhythmic and regular fluctuation in the calcium transient, and also showing that these cells do respond to adrenergic stimulation by epinephrine, and they show as a population of cells. So these do show the cardiomyocyte phenotypes. However, in our hands, we still really could not see beating cells. And so it shows that, at least in our hands, there's still improper organization of the contractile protein apparatus of this cell. So we call it cardiomyocyte-like cells. Eric Olsen in Texas, they have been able to show beating cells by this direct conversion pathway. So we continue to optimize the system. We also add low molecular weight inhibitors that would inhibit certain pathway and show that we can further improve this transdifferentiation pathway. We also look at different growth factors, again, with the right combination, we can begin to get better and better. So I am showing this study and to show that not all direct reprogramming is that simple. In this case, for example, with the transcriptome and the principal component analysis, we show that the phenotypes of these direct reprogrammed cells are still quite far from the native cardiomyocytes in the cardiac tissue, and also the cells are not as functionally mature as the cells derived from the iPSC source. So the summary for this, we show that there are a number of biochemical cues that we can apply for such direct reprogramming, but there is still a lot of room for improvement. Now, let me show the third example of the study. This is a little bit different using the CRISPR-Cas9, the genome editing approach to get the differentiated cells. I think many of you in the room are working on this genome editing approach. The CRISPR-Cas9 is one of the most amazing scientific breakthrough in the past few years that one can do genome editing in an efficient manner. So very quickly, Cas9 is an endonuclease, is an enzyme that has an epitope that binds to RNA, and it's about about 80 base pair and so on. So a guide RNA would be something like a 100 base pair where 80 base pair is constant binding to the Cas9, but then one can vary the sequence of the 20 base pair serving as a guide that could guide the complex to a certain size in the genome. And then once it gets there, the enzyme can cut the double helix. So this is a very powerful way to delete a certain gene. And if the gene of interest also happen to be in the nucleus of the cells, then based on the natural DNA repair mechanism, one could cut out the dysfunctional gene and insert the right sequence in there. So it's one of the most exciting way of doing genetic correction right now. So for this purpose, what we do is actually deactivate the Cas9. So this is an enzyme that will cut. It will not cut. So it mostly functions as a RNA binding protein now. Okay. So, but what we do is that fuse VP64 into the Cas9. And VP64 is a potent transcriptional activator that it would attract transcriptional factors to come into the site. So this will not cut, but this will activate the gene. And this is in collaboration with Charlie Gersbach at Duke. The intellectual force of this concept belongs to him. We were just lucky to be in the same building working with him and show that indeed with this kind of transactivation approach, one could activate different genes. We did activate the neuronal genes and to show that the cells could go into the neuronal lineage, but not really efficient at all. We can only see a few cells reprogram into neuronal cells. Shandon, a PhD student in my lab, independently hypothesized, well, maybe that we need to activate the gene to a higher level in order to get the reprogramming to work. So he proposed to fuse the VP64 to both the N terminus and the C terminus of this deactivated Cas9. And we did a lot of cloning in our lab with 3 NLS nuclear localization sequence. So that would bring this deactivated Cas9 back inside to the nucleus of the cell. Viral delivery versus non-viral. Of course, viral is much better, different guide RNA design and so on. And with this optimization, we can get beautiful myotube formation by the activation of the MyoD1 gene. We can directly convert the murine fibroblasts into the myocytes, and they could show multinucleated myotubes with great organization, and they also show intermittent twitching. So with that approach, even with human cells, again, with the same endothelial progenitor cells I show you, we can directly convert them into skeletal myocyte by this genome editing approach. And it is also a very efficient process. Now, then we can go back to the second to the neuronal transdifferentiation process that I show you now, using the deactivated but fused with 2 VP64 domain now, but not with one gene, with 3 different genes. We can show that we can activate those 3 genes, particularly at the site. However, this kind of genome editing approach is specific. So you would upregulate the gene at the site, but the overall expression level is still not as high as the ectopic expression, because now in the typical overexpression using a lentiviral vector, you can have the gene expression anywhere in the genome. But now we are activating only the genes at that site. With this approach, I'm just showing a lot of work in one single slide here. We can directly convert fibroblasts into functional neuronal cells. And these are the electrophysiology, single cell patch clamping, and so on. So this is still in the murine system. The other 2 systems I show you are the human, and so we still have to prove that this could be done in the human system, and this would be more difficult. But this shows the promise of this. So the summary of this transdifferentiation pathway is that using the CRISPR-Cas9 technology is also possible to directly convert adult cells from one lineage to another lineage. Now, you might ask why the CRISPR-Cas9 approach may be better than the typical overexpression ectopic approach. We are still doing this. It may be a more precisely controlled manner of doing it, but there are some practical advantages of this. For many differentiation pathway, in order to get the target cells, for example, if one wants to get functional motor neuron, in the early days, you need to deliver 7 genes in order to convert fibroblasts into functional motor neuron. Right now, you need maybe only 5. It's messy. You need to deliver 5 different viral vectors to a cells, and it's kind of a trauma to the cell. And in order to get that target cell, with this genome editing approach, I only need to deliver one single viral vector, or one vector that would express the deactivated Cas9. then I would deliver a bunch of guide RNA to activate different genes. So there's the ease of multiplexing and also the ease of co-delivery of the genome editing elements. And that's what we are doing right now. So there is tremendous promise with this kind of stem cells. On the therapeutic side, of course, for regenerative medicine, one could get this kind of tissues for therapeutics in vivo, and one could also do cell-based therapy to do gene correction to deal with genetic disorders. Also, tremendous value to gain fundamental insight on how disease develop, and particularly the genetic diseases. In the more near term, though, there's a great opportunity of using these cells for drug discovery. As I was showing you, giving you the flavor, now one could make this human cell-based, physiologically relevant model to do drug screening, and also could impact precision medicine, and may change the thinking on how drug development and also drug treatment could be done in the future. A number of researchers in the field, for example, such as Joe Wu at Stanford, they have advocated this concept of, for example, of clinical trial on a dish. And this is possible right now, and that's what we are doing. You can make hundreds of these iPSC lines from different population, and then you can generate library of these tissues for drug screening. So, to summarize, I have shown you that by delivering a different kind of biochemical cues, one can directly convert adult cells right now almost to any kind of cells that you would like to see, almost. Using overexpression of transcription factor, microRNA, and also low molecular weight, and also genome editing elements to do it. There is a lot of opportunity for us as biomedical engineers, as well as biomaterial scientists, to contribute to this exciting field. One, for example, is on the delivery aspect. The dream of regenerative medicine, in a way, or cell reprogramming, is to do in situ reprogramming. If, and this is a big if, for example, one could inject nanoparticles delivering these reprogramming factors into the brain that could convert astrocytes into dopaminergic neurons and so on. That would be the impact, and that would be the dream in the future. And that one would need to do a non-viral approach, and not just ex vivo. So there's a lot of challenge as well as excitement if one could optimize the delivery of those factors. One still need a better intracellular delivery of the transcription factors, as well as the intracellular delivery of genome editing elements. There's also the opportunity to optimize the microenvironment for the cells to reprogram from one lineage to another lineage. We know cells culture in 3D would behave very differently in 2D, very little work has been done in this reprogramming process in 3D, but there's a lot of opportunity. In the first example I show you is kind of a 3D on how endothelial progenitor cells under flow can be reprogrammed into the smooth muscle cells. We know mechanical kill would make a difference, and work has been done. However, very little work has been done on how electrical kill could promote this reprogramming cells from one lineage to another lineage. We have heard exciting talk from John earlier this morning, and those would be the technologies that could be integrated to advance this field, particularly in situ, in vivo, how they could drive the cells from one lineage to another. And there are other kills, such as topographical kills. We have done a lot of work in that area. And also ECM composition, which is not crowded, but make a big difference. And also the substrate compliance, probably many of you know, cells, stem cells, culture on a rigid versus a soft substrate makes a big difference, and they would go into different lineage based on the mechanical properties of the substrate. So these are all physical cues that could make a big difference, and we can optimize the microenvironment. Intellectually stimulating is probably also how can we dynamically modulate this microenvironment as cells transform from one lineage to another lineage, and this can be done. But how to do it in a robust manner, and again, in situ, in vivo, and that would be a challenge. The other aspect is the systems biology aspect, using that to look at the gene regulatory network and think of a better pathway to reprogram cells. And this is something also for the future, how to bring in the biomanufacturing considerations into this process. So there is ample opportunity for us as engineers to contribute to this exciting field. With that, I would like to thank all my students, fellows, collaborators to do that, and I would be happy to answer any questions. Thank you. Electronic portion, right now, very few people are incorporating that into the system. It's still flow and a 3D tissue, but eventually one would like to see online sensing. So it's kind of a future concept. Well, someone is actually doing it, but not at the manufacturable fashion, but it will have to be at that level for it to be impactful. That's why I say biomanufacturing would be a critical barrier for the translation of such technology. Please.
If I take any of the 206 cell types and I say I want to convert this cell into that cell, I have all the various options that you have outlined, CRISPR, MIR, viral, non-viral, and RNA. But your very first example was just one transcription factor.
Yes.
Myoglobin,
Myocardium, yeah.
which converted the endothelial molecule to muscle. So I'm wondering, do we have a sense of the complexity of that? Meaning this one was very simple, and some of them may be very complicated. Do we have some sense of which ones will be complicated and difficult, or which ones will be simple?
That is a great question. That's the systems biology aspect, and that's my purpose of showing the second example. Getting cardiomyocytes is very difficult. And hepatocytes, which is also doing, many groups are doing. I can take 20 ml of blood from any patient, convert them into hepatocytes, and to make a 3D tissue functional micro liver tissue. And that is intermediate, not so simple. And so you're asking, is there a theoretical framework that I can predict? I don't think we are there yet. It's still quite empirical. Again, that's an aspect that we engineers in the long run can make an impact.
Thanks for the nice talk. I was wondering if you can say a few words about how to go from here to towards maybe 3D printing of objects.
Yes, that's more at the tissue biofabrication level. For any kind of those organ tissue on a chip, eventually probably would need 3D bioprinting, because for drug screening, cost would be a big factor. You need to be manufacturable, high throughput, and that's where I see bioprinting would make a major impact. I don't know, I hope that answered the question. Also, 3D bioprinting would help multicellular tissue development, because in principle you can have precise organization of these multicellular structures.
On the issue of the drug discovery or the drug testing drugs with something like this, how do you deal with this issue of genetic diversity, or how would you program the genetic diversity into those cells that you did, and how important is it to do that?
Well, I was going a little bit quick, but this is actually a excitement. What I meant by clinical trial on a dish, you can get hundreds and slowly build up to maybe thousands of patients with different genetic population background and use that library for testing.
So you're saying probably the genetic materials in the blood cells that you started with.
Yes.
That could be for a particular patient, and so that's how you would get the diversity?
Yes, that's actually what we are doing right now. Yeah. We get, for example, the blood sample from Marfan's syndrome patient, make a blood vessel out of it and look at the vasal activity of that blood vessel. Please.
So in thinking about transitioning this technology, the liquid on a chip, tissue on a chip, towards kind of robust and widespread application in drug screening and drug development, to what extent do you think that the barriers to accomplishing that are known, and engineers and scientists just need to develop technical solutions to those known problems, versus what portion of the solution has yet to be discovered, or what barriers have yet to be discovered?
I think one of the questions alluded to that is the high throughput biomanufacturing aspect. So let me back up a little bit. When this first started, I think this will be a discussion at the end of the day. You talk to medical doctors, initially they said, oh, no way, this is not going to work. You can never capture the functions of a liver. It's so complicated. But the big pharmas are accepting that right now. I don't need to get a micro liver tissue that could recapitulate all the functions, but just enough functions that would be useful for drug testing. Actually, Becton Dickinson, for example, they are selling human hepatocyte spheroid on a dish, but those cells are not good because they are generated from cadaver and so on, and they do not have good functional characteristics. But now I think with this kind of microphysiological system, we can make it much more functional, and big pharma are accepting the concept that it could be done, that it could be useful. And I also emphasize the point that it's not going to totally replace the animal study, but just give someone a higher confidence of going into clinical trial. If you think about it, just a slight improvement could translate into a huge amount of saving in cost and time. And so the engineering challenge is still to make this kind of system as functional as possible, but to really make it high throughput and cost effective.
Okay, very good. So we will conclude this session with thanks to Professor Leong. And similarly, we have a gift for you, and I appreciate